Before I introduce the speaker for today's webinar, I would like to invite Professor Vajra Dissanayake, the current director of the Human Genetics Unit and patron of the Human Genetics Society, to share a few words with us before we start. So over to you, Prof.
Nirmala.
Okay.
I'd like to begin by congratulating the Human Genetics Society for having organized this inaugural webinar. As you quite rightly said, Nirmala, today is a very appropriate day for the society to start these webinars. Although we live in challenging times, it is necessary for us to continue to do what we are doing and continue to support continue to support our patient populations. The COVID epidemic should not result in others being left behind. So in that spirit, it's a good opportunity for everybody to come together on this DNA Day to reflect on what we are doing, the bread and butter of clinical genetics and genomics. And I hope that today's webinar would be the beginning of a long series of webinars, and that would lead to better understanding of genetics and genomics, not only in our country, but also in the region and beyond. And that would also create opportunities for everyone in this platform who are joining us to go together as a collaborative group promoting genetics and genomics and the adoption of those services based on genetics and genomics into our health systems. So thank you very much. I wish you all the very best with this series of webinars.
Thank you, Prof, for those encouraging words. Okay, so we will now proceed with the webinar. I would like to introduce the speaker for today's webinar. And she is Dr. Dulika Sumatipala. Dr. Sumatipala is a lecturer and clinical geneticist at the Human Genetics Unit, Department of Anatomy, Faculty of Medicine, University of Colombo, Sri Lanka. Her interests are genetics and genomic medicine with a special interest in neurogenetics. She is currently a PhD scholar in medical genetics at the University of Oslo, Norway. Her project focuses on exploring the pathogenicity of DNA variants in whole genome sequencing data in patients with unexplained progressive encephalopathy. So her talk today is based on her PhD research findings and is titled "The Use of Next Generation Sequencing Data to Diagnose a 20-Year Clinical Mystery." I would like to kindly request the audience to keep their microphones muted and videos switched off during the presentation. If you have any questions or comments, Dr. Dulika will address them at the end of her talk during the discussion time. So over to you, Dulika.
Thank you, Nirmala, as president of the Human Genetics Society, for actually inviting me as the inaugural speaker for the Human Genetics Society's webinars. So I thought I'd use this opportunity to speak about next generation sequencing and how it was used to solve a clinical case that had been in our files for over 20 years. So as Nirmala pointed out initially, this day is quite significant because of its importance, both as the day that the Human Genome Project was officially completed in 2003, and also because it was the date that Watson and Crick first described the DNA helix in 1953. So if we look at the timeline of genetic findings, we see that it had a first initial slow start with the discovery of the microscope and then the Mendel's law. You can see the time period between those two was around 300 years. Whereas if it comes to the 1953 and 1977, when the Sanger sequence was developed, there was only a 20-year gap between the two. And afterwards, the pace picked up quite rapidly. And the true innovations came to the forefront with the Human Genome Project, which was launched in the 1990s, and its completion. And in the early 2000s, a lot of companies came forward with next generation sequencing platforms, and the genetic diagnosis of diseases using next generation sequencing actually started off at a rapid pace from the early 2000s onwards. So if we actually look at how genetic diagnosis has been performed over the last couple of years, in this graph on the x-axis, we see the years 1986 onwards until 2016, and the phenotype, genes and associated phenotypes discovery on the y-axis. So initially in the early 1980s, conventional tools, karyotyping, arrays, HGS, etc., came into were the most used tools. But then we see in the 2010 onwards here, if you look at 2010, the 204 novel genes associated with phenotypes, we see approximately 13 were identified using next generation sequencing. And then we see the rapid pace in which diagnosis picked up with the use of next generation sequencing. So in 2012, there was almost equal contributions by next generation sequencing technologies and other conventional tools. But then it truly, there was a true increase in the rate of discoveries using next generation sequencing from there onwards. And up to date, I think we can safely say that next generation sequencing is the tool used for novel gene discovery and clinical discoveries for genes with associated phenotypes. So how does novel gene discovery look at when we take Mendelian phenotypes? So the human genome has approximately 20,000 genes. And if we take those 20,000 genes and graphically describe them, this is a 10 by 10 box graph. So altogether there's 100 boxes here. So we know approximately 4,000 genes are known to cause Mendelian phenotypes on OMIM. So 4,000 is approximately only 1/5 of all the known genes, 20%. So that's like the two bars here on top, known genes for a Mendelian phenotype. And everything apart from that is actually still to be discovered. So that's actually an optimistic overview. We've got a lot more discoveries to happen in the genetic world. So this is actually a great place to be right now. We have the potential to discover many more genes that cause Mendelian phenotypes. And if you look at the remaining boxes, there's two categories. There's the high priority candidates and the possible candidates. Possible candidates actually have no functional studies as well associated with them, while the high priority candidates have some functional studies which have aligned them with the possibility or probability that they might be associated with disease causation. And these probability predictions are based on both in vitro studies as well as in silico predictions. If we take in vitro studies, these are the green. They have been, the variants in these genes have actually been shown to be lethal in the mouse model. So that would be a good indicator that they would also have the potential to cause disease in humans. And in silico prediction models are the loss of function constrained genes, gain of function constraint predicted, and missense constrained genes. What we mean by this is that I guess everyone knows that a loss of function is when a variant causes the protein's function to be reduced or actually abolished in some instances due to a variant. And therefore a loss of function constrained gene means, so they have a high PLI. So what does this mean is the probability is given as a statistical score. And this is obtained by looking at the expected variants in a gene with those that are actually observed in that gene. So if we expect a couple, like 20 plus variants in a gene, and I only see 1 or 2, that would be an indicator that that has a loss of function constrainment. That means a loss of function would actually be highly pathogenic, and therefore they're not seen in that gene. Likewise, gain of function, I'm sure you know, is when a variant causes an abnormal function in that gene. So, and then such proteins are usually evade nonsense mediated decay, and they cause a novel phenotype. So likewise, in silico predictions for gain of functions are also seen, as well as missense, where a single nucleotide change can cause an amino acid change, and therefore a change in the protein function. So all of these are in silico prediction scores that can be made based on softwares, etc., and statistical modeling, while the green is the lethal in vitro modeling done by mouse models or zebrafish models, etc. So this approximately 30% of the 20,000 genes have had some sort of study that shows that they are possibly associated with a phenotype. So these are the ones that we should actually be looking in when we do research, because they're already halfway there. So all we have to do is make a connection with a disease phenotype. So next, so what do we actually hope to do with next generation sequencing? What is in exome sequencing, we sequence the whole exome, that is approximately 20,000 genes. And the result is usually 10,000 to 20,000 variants in the protein coding regions. But then that's when the real effort begins, because how do we select that 1 or 2 pathogenic variants that cause the disease phenotype out of the 20,000 plus variants that have been noticed with the sequencing? And those variants are not just random, not associated with any function. These are missense variants, loss of function variants, known variants in known disease genes, etc. So they're very plausible to cause a disease phenotype. But we have to find that one that is the pathogenic variant, which is practically like trying to find the needle in a haystack situation. So one of the tools that we can use in such a situation is the allele frequency. So we have our single exome, which has the list of 10,000 to 20,000 variants. But along with that, we have databases filled with tens of thousands of exomes, where each and every position in the exome has been looked at and a frequency has been noted. So we can use those tools and harness that power to analyze our exome as well. So these databases that collect frequencies of allele frequencies, initially in the early part of the decade, we were focusing on the exact database, but later on, the past 2 years, the Nomad database has come to the forefront. Actually, the exact has deposited all their data into the Nomad, and now the Nomad carries allele frequencies, sequence data on approximately more than 140,000 individuals. So what we're doing here is we're predicting the frequency of an allele variant in the population. So since we can't sequence the whole population, we're taking a cohort and looking at them to look at the frequency of a variant. And of course, in such a situation, it's quite obvious that the larger the cohort is, the better the prediction of that allele frequency is, and the closer it is to the actual population allele frequency. And so this database carries 125,000 plus exomes and 15,000 plus whole genomes provided by principal investigators of various scientific projects. So most of these have been situated in the European areas. Therefore, the ancestry which is divided and shown also in the genome database. A lot of it is taken up by those with the European ancestry, but here in yellow, we can also see that there is a considerable contribution by the South Asian population as well. So, so this is one tool that is extremely important. If we go into the Nomad, we would see something like this. So this is for a variant on chromosome 1, and this is the position on that chromosome, and the variant is a change from C to T. So here we would see in the heterozygous state in various populations how the numbers are, and in the whether that position, the homozygous change of this variant is, and ultimately you get an allele frequency. And if you look at this allele frequency, it's like out of 100, it's 9.068. So we know that a polymorphism is anything that is above 1%. And that would make this not a rare variant, therefore unlikely to be disease-causing in our variant list. So if we had this variant in our list, we would probably discard it as a polymorphism. But if you go on to the right-hand side, a few bases upstream, chromosome 1 and 5488, again a C to T change, there we see in the heterozygous state, there's only one individual in the whole of the Nomad database with this variant, and there's absolutely no homozygotes. And the allele frequency is 0.0005. So this is a rare variant. So this change is rare. Therefore, it could be predicted to be pathogenic. And if this variant was in our list of possible variants of the 20,000, one of the 20,000, we'd probably keep it aside to look into further because of its rare frequency. So this is one of the tools that we use for next generation sequencing, variant analysis. The next is, of course, in silico pathogenicity scores, whether the variant is predicted by softwares to be pathogenic or not. So the change in the nucleotide, so there's various annotation methods for this. One of them is conservation. So every position in the nucleotide, every gene position has been mapped either in zebra. So if that nucleotide position has remained the same throughout the phylogenetic tree, then we're looking at something that is possibly well conserved and therefore important for the functioning. Whereas if some nucleotides have changed from the zebrafish to the mouse model to the primates, etc., then because the conservation is not well preserved, we could predict, these are all predictions of course, that that nucleotide position is not essential to be remaining the same, and a change can be tolerated and therefore not disease causing. Likewise, there's the variant itself. So if you find the variant is insertion or deletion that actually changes the whole nucleotide amino acids line, then that would be much more predicted to be much more disease-causing or pathogenic than a missense variant. Likewise, if we take a missense, there's various amino acids. Some are polar, some are non-polar, some are acidic, etc. So if that nucleotide missense change of the amino acid converts what was previously a non-polar amino acid into a polar amino acid, that would be much more considered to be much more pathogenic than if the amino acid change was, for instance, a leucine to isoleucine, which would be pretty much the same polarity of the amino acid. So these are some of the considerations that we take in, that are taken in the annotations and into the statistical probability prediction models that are used in the CAD score. And using all of this in the background, it ultimately spits out a score, which is called the CAD score. And based on the CAD score, we can actually evaluate or predict whether that variant that we're looking at is actually more to the pathogenic side or non-pathogenic side. So this is called the in silico pathogenicity prediction using CAD. So if we have rare variant, so out of the 10,000 to 20,000 variants that we initially had, if we filtered it based on the frequency of the variant and have taken only those that are rare, and have also taken those rare variants and further filtered on the pathogenicity or the in silico pathogenicity based on something like the CAD score, we have a much smaller list, which we can actually work with and look into various databases, softwares. We here are some of the scientific literature are deposited in PubMed, Google Scholar. So all of the new research, scientific literature, and data on a gene's function is deposited into these various databases, and we can individually go into them and see what the gene's function is. And if the gene's function is somehow correlating with what we see in the disease phenotype, for instance, if it's a ciliopathy or if there is some neuronal function that has been seen, if there's some difference in the zebrafish model associated with variants in a certain gene that is in our list, etc., they become much more significant for the possible novel gene that causes the disease phenotype. So once we have that, we have basically done most of the work that is there for predicting or finding a novel disease-causing gene. But as a whole, if we look at it, how do we make next-generation sequencing truly effective in our research setting? The most obvious is, of course, doing as much exome sequencing or next-generation sequencing as possible, and to recruit as many families as possible, because if we have larger numbers coming in, of course, the diagnosis from the other end is also going to increase, not only because of the expertise improving, but also our knowledge regarding softwares and databases also improve, and our ability to do this filtering also improves. So recruiting patients as much as possible from clinics, making clinicians aware of rare possible syndromic diseases for the next generation sequencing studies is one of the most obvious methods to make a novel gene discovery. For instance, from a single exome, we know that there's approximately 30% chance of making a diagnosis. So if we have 100 cases, approximately 30 on the first run itself have the possibility of getting a definitive diagnosis. And what's interesting is that once we have next generation sequencing data in our hardware stored somewhere, we can come back to them 1 year or 5 years later and do a reanalysis on that same data using newer pipelines, newer softwares, newer databases. And this reanalysis actually has been shown to improve the diagnostic capacity to a further 10 to 20%. So in total, we have the possibility of actually doing up to 50% of a diagnosis per case. Whole genome sequencing is also a possibility. In the clinical setting, of course, it hasn't been as widely used, mainly because research studies have actually showed that they improve upon the diagnostic capacity by approximately 5% alone. So this cost-benefit ratio is still weighed against using whole genomes in the clinical setting. But of course, in the future, when prices go down on sequencing as it is been showing in the last few years, then there is a possibility that genome sequencing also might come into the clinic. But at least now, for now, whole exome sequencing seems to be doing an adequate job. So following that method, first initial, most obvious method of doing as much next generation sequencing as possible. The second most effective method of novel gene discovery is actually data sharing. So once you've identified your candidate gene, finding another family, unrelated second family with the same gene, having a variant in the same gene with overlapping phenotype is actually the most effective method to taking a candidate gene into a confirmed disease gene and to a research publication. Because these are rare diseases, the possibility of finding one within your own study is quite rare, but if you can look around the whole globe and find some other family with a similar gene variant, that's pretty much closes the whole case for most instances. So what are the platforms that are being used for this? GeneMatcher is actually the most well-known platform used for data sharing. Here we see the graph where researchers have used GeneMatcher. There's an exponential rise in the usage of this tool. And what is done is actually, if a researcher or clinician has a specific candidate gene, there is no known association with a phenotype. However, it looks very promising as a candidate gene. They deposit it into GeneMatcher, and the GeneMatcher actually makes a connection with all the deposits within it and looks for a similar gene, a variant deposited with a similar gene. And they actually just share the email addresses of the two research groups. And then you can make the connection, see if the phenotypes overlap, and then just go onwards with the research and publication. So GeneMatcher is one of the most commonly used, but there's also some other platforms like Matchbox and MyGene2. So these are all data sharing mechanisms and is actually one of the fastest methods to get a candidate gene into a definitive disease-causing gene state. So if both of these methods, if you can find a variant that is actually looking promising, and you've stuck with a good case with a syndromic phenotype and you don't know what to do, then it's time to look at alternative methods to using that next generation sequencing data that you have in hand. So we, some of these alternative methods include shifting from the SNP indel variant pipeline to a structural variant analysis pipeline, or using various tissues like fibroblasts, muscle, etc., to do transcriptome profiling to see if there's some mosaic or RNA, mRNA-based disease tissue gene expressions, or if there's an epigenetic change, something like chromatin immune precipitation and sequencing, which looks at transcription regulation and gene expression. So these are some of the methods that are used beyond the mainstream SNP indel pipeline. So actually my clinical case will be featuring the structural variant analysis pipeline used on next generation sequencing. So this case was initially seen in 1995 actually. The patient had developmental delay with hypotonia from birth, and some milestones were achieved, speech and gross motor. But from the age of 2 years onwards, the patient had had severe regression, generalized tonic-clonic seizures, and neuropathy with muscular atrophy, respiratory failure, and had died at the age of 11 years. Now, this patient had one other sibling. The parents were not known to be consanguineous. And so this patient actually has been published as a clinical case as a novel, possible novel syndrome in 1997 as well. So what happened was a pathological autopsy was performed, and what was striking at that autopsy was that despite the brain having atrophy, the weight was within normal limits. So then the first clue to this being a storage disease was made. And with that, they checked the lysosomal enzyme activity using skin fibroblast of the patient. And all of the enzymes had actually been showing normal activity. So what do they do from there? They did the electron microscopy of the motor neurons of the spinal cord. And what was seen was this stacked cytoplasmic region, which is also called zebra bodies. We can see several here on this image. And furthermore, they had gone with UV light based light microscopy, where they had seen autofluorescent, these bright red storage material seen in the cytoplasm of motor neurons. And immunohistochemistry had also been done, and they had done it for GM2 ganglioside, and a known GM2 ganglioside disease is actually Tay-Sachs disease. And in the presence of GM2 ganglioside, you get this red autofluorescence. So this is actually a positive control, and this is a negative control without the red autofluorescence. And here we have the patient's motor neurons with the red positive indication that the patient had GM2 ganglioside storage material. So in 1997, so a pretty much complete phenotyping was performed, which I'd also like to highlight as something that is very important to come to disease gene diagnosis, having a clinical picture that is as complete as possible. So here we had a pretty good clinical picture, a lysosomal storage disease with GM2 ganglioside accumulation. And in 1997, they had hypothesized the genetic defect is most likely. So over the span of the next 20 years, pretty much every possible genetic test was thrown at this patient's patient material, karyotyping, array, whole genome, whole exome, sequencing, etc. So once the SNP-indel pipeline also gave no findings that were compatible with the disease phenotype, they went on with the structural variant phenotype, structural variant pipeline analysis in 2017, so 20 years on. And what was found was that there were 2 deletions, homozygous deletions in a gene called TBCK. So these were massive, massive in the sense they would not have been recognized in the SNP-indel pipeline, but they were also not large enough to be noticed on an array. So a usual diagnostic array has a limit of a size around 25 kb. So this was 7 kb in size. So it really fell in the middle of diagnostic capabilities. And what we, this 7 kb deletion took out exon 23 of the TBCK gene. So it takes out a whole exon. So obviously that would mean several amino acids being deleted out of the protein, most likely the protein going into nonsense mediated decay. So the effect on the protein function is massive. And also it has never been previously noted in any of the copy number variant or structural variant population databases, and all in silico path prediction tools gave it as damaging. So it was a very likely the cause of the disease in the patient. And here we see the sequencing data on the IGV tool, so integrated genomic viewer. So there's complete loss of the sequencing data in between the region of the deletion. If you look at the parents' data, the mother and the father, you can see it's a bit difficult to actually notice, but there's a 50% drop in the number of reads in both parents, so that both parents were heterozygous carriers of the deletion. And so the genome sequencing finally gave the cause of disease to be due to a homozygous deletion of TBCK. So next we went on to the scientific literature and TBCK encephaloneuropathy, and the clinical phenotype overlapped quite well with what was seen in the patients, with the developmental delay, respiratory functions diminished, and also the distinctive faces. So altogether, the clinical phenotype overlapped, and the pathophysiological mechanism was the next question. We had storage material in the interneuronal cytoplasmic cytoplasm of the patient on autopsy. And what TBCK does is it's an inhibitor of the mTOR pathway, and the mTOR pathway is known to be associated with autophagosome flux. So the loss of function of TBCK actually increased the autophagosome flux to such a degree that the acceleration caused inefficient degradation. So there was nothing wrong with the lysosomal enzymes per se, but this acceleration of autophagosome to autolysosome creation was such that everything within the autolysosome was being inefficiently degraded, and you got membranes stacked one upon another. So the clinical picture was pretty much well explained by the pathophysiological mechanism of the loss of function of the gene. So it was an interesting study, and it was an interesting utilization of next generation sequencing. And so this is actually my last slide. What we can take from this talk is that genomic technologies are transforming our ability to discover the genetic basis of rare diseases. There's still a huge scope left in the discovery aspect of OMIM genes, and there's still tens of thousands of genes left for us to discover. And as a community, as a research community, one of the best ways is to empower our data sharing so that we can connect with one another and discover the remaining genes and what is the underlying Mendelian phenotype that is caused by variants in these genes. And the tools, we have the reference population databases, which are critically important for us to continue on these studies. And also, since with the databases, the analysis softwares are also being updated almost daily. So an important aspect to remember is to keep on going back to previously negative sequencing data and reanalyze them and increase the diagnostic capabilities. So that's all, I think. Yes. If you have any questions, I'll be happy to answer them as well as I can. But yeah, I think I'll hand it over to Nirmala now.
Okay, thank you so much, Dr. Dulika, for that excellent presentation. The floor is actually open now, so if anybody has questions or comments to make regarding the presentation, you're welcome to do so.
Hi, Dr. Dulika. Thank you. That was a very thought-provoking presentation. I just have one question. Now, when you find the structural variant in this TBCK gene, and in silico, there was a prediction that it was deleterious and also the phenotypic features matched it. Was there any place for functional studies, like on a zebrafish model, or to look at how it's going to affect in a biological model?
Yes, there is always the potential to do functional studies. Even the patient's skin fibroblast and doing immunohistochemistry for autophagosomes, those are also compatible. And also for the mTOR pathway, if you look at the various components of it, so there's increase in the function of the mTOR pathway, so you can do protein studies to detect whether they are increased in comparison with controls. So a lot of them, I think one of the slides I had actually given the reference to one of the research groups that had done a study, a functional study. So these are some of the studies that you can do. So once you know, once you have the clinical phenotype, autopsy material, and have some background on the function of the genes. So this, I have a slide now. So these various components seen in the mTOR pathway, such as so ATG13, etc., they can be calculated and quantified, and therefore compared with patients and controls. And
okay. In this pathway, it's probably because the secondary metabolites are not degraded properly, right? So if we look at the secondary metabolite levels in the patients, is that enough? Or,
yeah, I think you could do metabolic studies on secondary.
Yeah. And I have another question, sorry. So now when you look at like new gene discoveries, like for example, we have already looked at all the genes that you can think of, and now you want to see whether there's something that you're missing. Do you go back using the CADSCO and you look at all the like the damaging mutations and then you look at the genes, or how do you do that? What is
I usually like to start with the frequencies, because if you go on Nomad and see absolutely nobody with a homozygous mutation, or like nobody with even a heterozygous variant in that position, then that's a very rare variant, and that is the most I think significant, because the Nomad is so large now. It's actually more or less quite close to, I think, population frequencies. So that means your case has actually the one and only in the world, something like that. Right. So that's very significant. And then from that, it actually cuts down the list to quite significantly, brings it down to 100 or 200, etc. And from there, of course, the CAD scores are important, and looking at scientific literature, because going into scientific literature, you might know it's a bit like going through the rabbit hole. It's like more and more papers about every single aspect of genes are being published daily. So yeah, it's a lot to read. So if you have some method to cut down your list to as little as possible, then use those first.
Okay, thank you.
Yeah.
Okay, any more questions from the audience, please? You can unmute your mics if you want to speak, please. Okay, just to inform that we will have the recorded version of this webinar, and we'll be uploading it to our Human Genetics Society website, probably early next week. If you want to listen to it again, you can always download it from the archives in our website at humangeneticsociety.lk. So you're always welcome to visit our site and go through the other contents there. So in the absence of any more questions or comments, I would like to once more thank Dr. Dalika for that brilliant presentation and for educating us on the value of NGS and how it can be applied in the clinic. And I would also like to thank everyone else for your active participation in today's webinar, which was our inaugural. So definitely we will continue with this. Every month we will be holding an academic webinar, and we'll keep you informed. The next one will be sometime maybe in 3-4 weeks time, and the details would be communicated to you in due time. So thank you very much, everyone, and we'll see you next week. Sorry, next month.